Hello everyone, welcome back to Dr. K. Primer lecture series presenting by Dr. K. Prem, that's me. In today's lecture, we'll talk about what is restriction digestion, then the difference between the linearized DNA molecule digestion and circular DNA molecule digestion, what is single and double digestion. These are the things we will talk in today's lecture. But what is restriction digestion? In simple way, fragmentation of DNA molecule by using restriction endonucleases is termed as restriction digestion. The number of fragments you get after the digestion is depends on the number of restriction sites present in the given DNA molecule. But the shape of the DNA molecule also determines the number of DNA fragments released after digestion, right? So in the next slide, we will see the differences between the linearized DNA molecule digestion and circular DNA digestion and the single digestion. Then followed by, we will see the double digestion. Here you can see it's a double-strand DNA molecule where it has eco and restriction site in the left side, which are bolded. And during the restriction digestion, there are 3 steps. First, when you incubate the double-strand DNA with the restriction endonuclease, the restriction endonuclease scans the given DNA molecule for its recognition sequence. That's a scanning. The second step is binding to the recognition sequence or restriction site. Third step is cleavage or breaking. So during the restriction digestion, you can see these 3 steps. The same thing here I am depicted. You can see single digestion of linear DNA molecule. Scanning. So ECORN enzyme will scan for its restriction site. Once it identifies its restriction site in the given DNA molecule, it binds to it. The second step is binding to recognition sequence. Then third step is cleavage. Especially type 2 restriction endonucleases tend to cleave within the restriction site or recognition site. They may be the cohesive digestion or they may release the cohesive ends or blunt ends. But restriction endonuclease, which belongs to the class 2, will cleave within the restriction site. You can see the cleavage here. Once it cleaves, cleaving means the phosphodiester bond, which is present in the, which is connecting the 2 nucleotides of the strand, is going to be break down in the both strands. Then it releases the DNA fragments. You can see here. Whenever DNA fragments which are released having the overhang, means single strand nucleotides, right? Those are termed as a cohesive ends. You can see. So when a double strand DNA molecule having the single restriction sites, after digestion, you can get the 2 DNA fragments. That's a linear one. When you compare with the circular one, you see single digestion of circular DNA. Here substrate is in a circular shape. So, and having only one restriction site. That's again a GAA TTC. As I told you that during the restriction digestion, enzyme scans the template or substrate and binds to the restriction site, then followed by the cleavage. These are the 3 steps happens during the restriction digestion. The, irrespective of the whether the DNA is a linear form or circular form, these three steps involves in the restriction digestion. You can see here, EcoR1, the circular DNA is incubated with the EcoR1 enzyme, and it search for restriction site. Once it identifies, it binds the restriction site, then followed by cleaving. Cleaving of two phosphodiester bonds in the two strands and releases the DNA fragment. Here, you can see one restriction site is there. The circular DNA becomes linearized DNA with one digestion. Whereas linear DNA molecule, when it is having one side, it releases two fragments. But whereas the circular DNA molecule, when it is having only one side, that is linearized. So the linearized DNA molecule can give the DNA fragments depends on the restriction site. If it is having one restriction site, it can give one more, means number of restriction sites plus one extra fragment. We can get during the linearized DNA fragment digestion. But when it comes to the circular DNA molecule, the number of DNA fragments are exactly same as number of restriction sites. So here you have only one restriction site, you got the only one fragment. But in case of the linearized DNA, it is having one restriction site, but two DNA fragments. This is the difference between the linearized DNA molecule restriction digestion and circular DNA molecule digestion. You can see. So linearized DNA molecule is able to give two DNA fragments with the one restriction site, whereas the circular DNA molecule having one restriction site give only one fragment, right? So here we have used the only one enzyme, hence it is termed as a single digestion. You see, what is the enzyme you used here? EcoR1, right? EcoR1. So this digestion is called as single digestion. In the next slide, we'll see the double digestion. You see, this is a double-strand DNA molecule having the two sites. One is EcoR1, GAA-TTC. Other one is the small one, CCC, GGG, right? So it's a double-strand DNA molecule. Now, when you incubate it with the two enzymes, and the two enzymes look specifically for their recognition sequence, right? Once they identify their recognition sequence, then they'll bind to that, and then followed by the cleavage happens, right? We are calling double digestion because the substrate is incubated with the two enzymes, and they cut within their restriction site, not outside their restriction site. You can see the same thing here. First is scanning. EcoR1 goes to its respective restriction site, binds to it, right? Then SMA1 also scans for its restriction site and stops at its restriction site. That's a CCC GGG. Then this is once it stops at its restriction site, that's called as a binding to a recognition sequence. Then third step is cleavage. You can see cleavage. So EcoR1 cleaves at the cleaves and gives the cohesive ends, whereas small one exactly cleaves in the center of the restriction site and gives the blunt ends. See? So the EcoR1 side, after EcoR1 digestion, you can see cohesive ends, means a single strand overhang is there. But SMA1 digestion leads to the production of blunt-end DNA fragments. Blunt-end DNA fragments. So this duplex DNA, this double-strand DNA have two restriction sites. Hence, it can give 3 DNA fragments. As I told you that linear DNA will get 3 fragments, means the number of DNA recognition sequences plus 1 extra fragment, right? Here, you use the 2 enzymes for the digestion of same DNA fragment. Hence, it is called as double digestion. Double digestion usually employed in screening of recombinants. Whenever a clone is having an insert, which are cloned with the help of 2 different enzymes, then during the screening, you isolate the plasmids and go for double digestion. If you can that plasmid is able to release the DNA fragment, that means that's a recombinant molecule. So without wasting much time, right? So you can do the double digestion when they have a compatible buffers. When they have a compatible buffer, 2 enzymes can employ during digestion and releases the insert molecule from that clone. So this is all about the single digestion, double digestion, digestion of the linear molecule and circular molecule. And hope you like this content. If at all you like it, try to subscribe my channel. That's a Dr. K. PREM-PRIMER. And if you have any comments, let me know through the comment section. I'll get back to you as soon as possible. And if you want to, you don't forget to give thumbs up if you like it. Thank you all. Right? Fragment 1, fragment 2, and fragment 3. Thank you all. See you again. Until then, bye-bye.